What have you in your hand there?
This is, weirdly, this is my biology notebook from 5th year in school.
I can't believe you actually still have it.
Well, yeah. I have no idea, because I never throw anything out because I'm a hoarder. I don't know, but my kids think that all of their worst judgments on me are absolutely true, that I'm a complete nerd.
Well, when you look at that, it's pretty
nerdy.
It's really pretty impressive. It looks actually like an illustration in I just put it this way. It looks like an illustration in a biology book, not to mind it, just your biology school copybook. So did you take these notes actually in class?
No, well, I just must be really sad because I see it says on the spine additional biology notes.
Oh my gosh.
So yeah, so my kids, my kids may be correct, but I think it shows a love of detail.
But yeah, you must have been. This is the muscle, isn't it?
Muscle contraction. I'm not sure how much more I know than what I knew then,
well,
which is kind of funny.
I wonder, was this even stuff that you were actually taught in class, or were you reading stuff around? I think you must have been. It
possibly.
Seems really detailed.
Yeah, I certainly didn't use bullet points. No. It's all full sentence. Yeah, and I definitely don't remember anybody ever knowing the biology of the tooth.
No, I don't either. I mean, I did study biology. That's what I was thinking when I looked through it first.
Look, it says the number of your different from the centre over, how many teeth you have, permanent teeth.
So funny.
Yeah, it's weird.
But did you go into this level of detail in all the different subjects, do you know?
Yes. Biology, chemistry, history, English, yeah, probably.
And do you still have all of those as well?
They may be in my mother's attic.
Okay.
But this was actually in my own house. I have my physics one and my chemistry one. But what's kind of interesting is the physics one is really miserable looking, and there are loads of things drawn in the margin and stuff. I didn't get that. No.
You weren't whipped.
But what's really funny is they all refer to the Christian Brothers book and which page on the Christian Brothers, because it was a Christian Brothers physics book which was red and a Christian Brothers chemistry book which was yellow.
There was that level of sort of rigour already.
This refers to, there was a biology book called Roberts that we used to use for, if we were really interested in stuff, and it's in there. I mean,
that's actually bonkers.
Well, not really. I mean, there wasn't much else to do in Ireland at the time. Like, it was fuel rationing. We'd Angel's Delight and boiling the bag ham,
Exactly.
It was a different time.
Did you eat Angel Delight? I used to love Angel Delight.
Straight out of the packet without adding the milk.
Well, I think I might have done that too. And I used to eat jelly without making the jelly.
Yeah.
Bad habits.
Yeah.
So, yeah, we have such an unbelievably interesting a range of objects in front of us here. Do you want to maybe introduce yourself, and then we'll start talking about some of them?
Just one thing I will say about the physics copybook, which is really extraordinary. So it has a list of, like all these experiments that you did, to measure little g and gold leaf electroscope, all this stuff. What's unbelievable is I found my granddad's physics copybook from the Christian Brothers in Carrick-on-Suir from 1920 something.
It's in the family.
No, he did exactly the same physics stuff and had exactly the same level of disinterest as I had, judging by the state of his copybook. But isn't it funny, the curriculum was exactly the same.
That's incredible, but also incredible that you found it, his copybook, that it was kept. So there's obviously
hoarders.
Or a value on
material things, yeah. I mean, it goes without saying I have every single birthday card. I have every single letter. I moved to America in 1988 and I have the San Diego letters. And it's kind of weird, but I was going through them and I found these letters, and it was about a close friend of mine from my school days, and she met this person that she subsequently married. She used to write like 8 A4 pages of a letter. And I've all these, and suddenly I realise I've all these insights into other people's lives that aren't my life. And actually, I'm not breaking off the friendship, but I need to just send them back to her and to other friends that sent me these things. Yeah, it's kind of weird to hold on to other people's lives.
I've thought about that before, that when you send a letter, I've done that, I've kept letters, and I realise I only have one half of this conversation, and the part that relates to me, I don't have it.
I know.
It's very strange.
But I think it's kind of a little bit wrong to have this level of insight into people that belongs to them and not to you, if you know what I mean.
I know, it's a funny one.
Oh yeah.
Yeah.
So I have packaged up things,
Well,
not quite with ribbons around them, but
I have to say, when you are doing your biography, I think the San Diego letters should be a chapter. It sounds
it's
like
pretty.
a chapter
It's
of a book, doesn't it?
well, I'll tell you, it might be the entire volume 1. There's a lot in it, and I have all of the baby cards and all of that stuff. 3 kids, 3 lots of baby cards, every birthday card. It's just.
Oh my goodness.
Yeah.
And do you actually have a dedicated space that you keep all these things?
Yeah,
Or
and they're
okay.
filed. Well, there's on
You
top
have
of the
archive
micro.
already prepared. Like.
Well, there's the on top of the microwave in the kitchen bit, and then there's actually the boxes that say letters, photos.
Amazing. Amazing.
Well, no, I mean the photos are a bit messier. It has to be said.
Yeah. That's actually really nice, though. Well, I think it's really nice. Your children might not, in the same way that my children might not. But I think it's really nice for them that they have these things. You know, that, like, I'm really bad at taking photographs of my children, and I've moved house so many times that I just have one absolutely chaotic box that they can never find the photograph they want. You know, so it's,
Yeah, but I handed my husband a photograph the other day. It was just like an ordinary photograph from the times when you used to print photographs, and I handed it to him. And this is truthfully what he did. He did try and enlarge it.
yeah.
We're so used to like.
Absolutely, yeah. So, listen, introduce yourself there.
I'm Catherine Godson, and I'm a biomedical researcher at the Conway Institute, UCD.
Okay, so that's why we have some things here on the table that look really quite sciencey.
Yeah.
Tell me, what is this? Almost every person I've spoken to has had a tiny vial of something on the table. So what's this?
So I am particularly interested in a kind of a messenger molecule that the body produces when there's inflammation. And why we're interested in this particular molecule is because it damps down the inflammatory response and it actually promotes resolution. So instead of inflammation persists, so we all need inflammation to defend ourselves, but instead of the inflammation persisting, several different kinds of molecules are produced that actually stop, for example, the blood cells trafficking in their tracks, literally. And one of the molecules that's involved here is a lipid called lipoxin. And this is, we buy lipoxins commercially, they're made commercially, and they're very labile, which means that they're very unstable. Right. And so they have to be protected, for example, from light or from air because they can oxidize. So we store them in these particularly tight vials, glass vials, and this is lipoxin A4. So this is 10 millimolar concentration of, or 100 nanomolar, micromolar concentration of lipoxin A4. And we store it so there's a special membrane on the top so that air can't get in. So when we're taking it out, we don't open the vial, we use a special kind of syringe.
Okay, wow.
And so then what we often do in our experiments is, so we take lipoxin out and then we add it to cells and we see what kind of effect it has on particular kinds of cells. And what we want to see is what does it do to cells and how does it do it? Because we always want to know more and more details so we can refine and understand better how it works. So an example of how it works is, so in this picture, in this here, can I?
Yeah.
Yeah.
Amazing.
So what we have here is, so if you think of at a site of inflammation, white blood cells have to rush in to protect the body from, say, from injury or infection. And the first kind of cells that come in response to signals that get released are neutrophils. So neutrophils come in and, for example, they release toxic material that can, for example, kill bacteria. So the next thing that happens is the neutrophils are basically spent after all this exertion and they die. But you don't want dead cells hanging around. So the next lot of cells that come in are another kind of white blood cells called monocytes. And when they come out of the blood to the site of inflammation, so out of the blood vessels, they become what we call a kind of cell called macrophage. And a macrophage, so what macrophage means is big eater. So what macrophages do is they hoover up the dirt at the site, and the dirt includes these cells, these neutrophils that have died. And so we know that when we add, what we found in our lab many years ago, what we found is that when we added this lipoxin stuff 2 macrophages, we were able to stimulate them so that they would clean up the dead the dead neutrophils at the site of inflammation. And so what we see here is, so the green is the macrophage, and the kind of speckledy thing is the skeleton of the cell. And so when the cell is hit, or when the cell encounters lipoxin, it provokes a specific reaction that makes the skeleton of the cell rearrange, so it's actually able to essentially eat up or hoover up the neutrophils. And so these 2 things here, so the blue is the nucleus, so this is the nuclei. So this macrophage is quite greedy, and it's eaten 2 neutrophils, and then this is its own nucleus, yeah. Wow. So lipoxins stimulate this process to happen. So what we are really interested in is understanding, firstly, how much lipoxin do we need for this to happen? What happens when the lipoxin engages with the cell membrane? What are the molecules that receive the signal? How is that signal then transduced? How does that signal tell the cell, change your skeleton? I read something recently about this process, and it was describing it because it's become a subject of focus of a lot of people doing research. And the article was called "How to Eat Something Bigger Than Your Head." So, which is because they're changing their shape, so they make like a kind of a cup, a phagocytic cup, so that the dead neutrophil can be ingested. Because if that doesn't happen, and the neutrophil hangs around, then it provokes a lot of inappropriate inflammatory responses.
Do you still find it absolutely incredible that all of this stuff is going on, all those things you've just described, this gets applied and how this works, how it gets transduced and how it gets this, and how all of these things are going on, and we don't perceive them?
What I can't believe is how rarely they go wrong. Like how exquisite this dynamic equilibrium between a cell being made, a cell dying, a signal coming, how the signals are generated at the right time. Because, I mean, you wouldn't want this signal to be generated when you didn't want an inflammatory response, because we know that amongst the things that this does is it actually stops, so for the cells to come into a site of inflammation, they've got to stick to the wall of the blood vessel, then the vessel wall has to become leaky and they have to be able to traffic through. So you don't want these things being produced at the wrong time. So how it doesn't go wrong more often is amazing. And I think that the whole idea that, like, it's like the dynamic equilibrium that your body's in, it makes me wonder why anybody would choose surgery, why you would choose to open it up and expose it to what could happen. I just don't get, I just think it's amazing. It's like, when you think of kids and the development in utero, like, so that the right things come on at the right time and then they switch back off. And we know that often, and in some of the work that we've done on diabetic kidney disease in particular, we found that often diseases actually, it reactivates these processes of, the embryonic, the developmental processes, so things that aren't normally there. And that's like the body's kind of trying to get back to re-engineer itself back to a more plastic state. And actually these macrophage cells are incredibly interesting because they're really plastic, and depending on the environment that they find themselves in, they respond to the different cues that they find. So, the signal could tell them, be aggressive, kill bacteria, or it could say, actually, calm down, clean up the neutrophils, repair, regenerate. They're really, the cellular plasticity is amazing.
I find it incredible. I think it must be an amazing thing to be working in and actually discovering things and finding things out and having an influence, writing these papers, and getting a chance to just
make discoveries,
and also to sort of experience this wonder again. It's like the fact that you don't lose that sense of.
Well, the thing that's the real privilege is, it's not what you do, it's what you train other people to do. And it's your legacy is not what you do. It's not this, it's the people that you train that go on. And, you know, it is the continuum, it is the
Which is a
shoulders of
particular thing about
giants.
science. It's a
It's
particular thing about
amazing,
science
yeah.
that isn't
It's
there.
an iterative
It's not in
process.
art. It's not in art, for
Yeah.
example. I mean, I'm an artist. I'm Lorna Donlon. I was artist in residence in the Conway Institute from September 20 to September 21, which is why I have had an opportunity to meet with people like yourself and to talk to you about these things. You don't get that in art. You know, I love the way a paper comes out and the scientists go to the methods. You know,
Yeah.
and like, how can we get to do this?
The
How
first
do
thing,
we replicate this? How do we do it?
First thing I look at are the figures to see, like, what do they actually find?
Yeah.
But actually, the reason why I have these, so I did my PhD in UCD, then I went to San Diego. So when I was a graduate student, there was a journal called the Journal of Biological Chemistry, and it was really it was the gold standard. It was the New York Times. It wasn't Nature, which is a bit flashier, but it was really, I used to look at Dr. Beery, that was one of our lecturers, going through JBC, and I used to think, oh my God. And then I went to San Diego as a postdoc with the training I had got in UCD. And suddenly I was publishing a paper in JBC, and that was really thrilling. But then I came back here after lots of years abroad, and then from UCD, we were also publishing papers in JBC, and that has gone on, and that's really interesting, because for scientists, like, local is meaningless. It has to be international, and it has to be that other people all over the world not recognize you, but recognize the work that you do. Exactly. And one of the really gratifying things is when you find, maybe 3 or 4 years after you publish something, you find by just coincidence that somebody else has found qualitatively or quantitatively exactly the same thing. And often they don't even bother referring to your work, and you realize you have contributed in whatever tiny bit to the truth, because it's all about looking for the truth. It's about searching for the truth. And that's really important. And however tiny it is, that's really exciting, I think.
Brilliant. I think we leave it at that. That's really cool.
Great.
Thank you.